Welcome, my fellow biochemists and biotechnologists or biotechs, right? So, this series of videos will be about BCHEM 563, or as some of you will know, BCHEM 565, okay, and specifically for MPhil Biotech and MPhil Biochemistry. This particular one will be on Module 1, which is our introductory lecture, okay. And before we start, please do not forget to do your term paper and do that before the deadline, okay, because doc is not going to, accept any form of excuses before. So what we are going to start with looking at what genetics is. Okay, so genetics is simply the study of the rules that govern the transmission or the transfer of genetic information. Okay, and this transmission of genetic information actually occurs at three levels. Okay, the first level is the transmission genetics, which is which happens within families. So transmission genetics will be the transfer of genetic material from parents to offspring. Okay, and the second one is at the molecular level, which is within and between cells. So molecular and developmental genetics, which is the second level, will be the transfer of genetic material from DNA to gene. Okay. And the last one or the third one will be population evolutionary genetics, which is the transfer of genetic material in many generations. Okay. So this actually has to do with evolution and stuff. These two tables here are just key events in the history of genetics. Okay. For history, I think you have to just get time and sit with this and have fun with it as well. So let's look at nucleic acids. So just like proteins and carbohydrates are, nucleic acids are also biomolecules. Okay, but what makes them stand out is their ability to carry or serve as carriers of genetic information. So unlike carbohydrates and proteins, they even though they are biomolecules, they carry genetic information. Okay, so beyond their structural differences, this is one of the main factors that differentiate between nucleic acids and other biomolecules present in the human in living organisms. Okay, so there are two forms of nucleic acids. We have DNA and RNA. I'm sure you all know this. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So basically, DNA is just it's actually a storehouse for genetic information. Okay, so it's a storehouse or cellular library containing all information necessary for building cells and tissues of organisms. And the RNA is just so RNA. We actually have types, and we are going to talk about those. But let me call it the functional or the coding RNA, which we are going to talk about it later. Mostly serve as a copied form of the DNA. Okay, so mRNA usually serve as something that stores a copied form of the genetic information from the DNA. Okay, so it's not actually a storehouse, but it just carries a copy of the information from DNA. So these are three distinct structures or rules of RNA. Okay, in protein synthesis, we have the messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA is the one that carries the instruction or the copied genetic information from the DNA, and the transfer RNA is what interprets information within mRNA and also bring the correct amino acid, okay, for protein synthesis or the polypeptide synthesis. And the last one is rRNA, which is ribosomal RNA, and is the most abundant form of RNA, and it maintains translational enzyme complex, blah 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 blah. So we are going to look at all of this as well, and there are some other minor or small RNAs. Example is the microRNA, siRNA, SNO, LNC, and SN. There are so many others, and we are going to discuss them as well later in our videos. Okay, so where in the cell our nucleic acid is located. So different levels of organisms have different locations for their nucleic acids. Okay, in high organisms or eukaryotes, we actually have our nucleic acid or genetic material in our nucleus. Okay, but in prokaryotes, for example, they do not have nucleus. Okay, so there is actually an area, okay, that's like it's not membrane bound. Okay, so if it had a membrane, it would have been the nucleus, but because it's not membrane bound, we can't call it a nucleus. That's where the nucleic acids are found. Okay, so we say the nucleic acids are boundless in the cytoplasm. Okay, there's actually a name for that region. Is it new new nucleus. I've forgotten. Sorry. Um. Also, besides the cell having its own genetic material, some organelles like the mitochondria and also chloroplast contain extra maternally inherited genetic material. Okay, so the mitochondria will have its own DNA. Okay, besides the cell having its DNA, and in plants as well, chloroplast will also have its DNA. And this DNA in these specific organelles are actually maternal genetic material, and they are not inherited from their father. Okay, so so what someone can ask this question: why is carbohydrates, or why are carbohydrates, proteins, nor lipids, or even RNA? Why are these not the genetic material but DNA? And how did we come to the conclusion that DNA was the genetic material? So these are we are going to talk about a series of experiments done. Okay, they've been done already, and how they established that DNA is actually the genetic material. So the first experiment was done by Griffith. Okay, and we call it the Griffith's experiment. Griffiths worked with two strains. Okay, two strains of viruses, the S strain and the R strain. The S strain is actually known to be infectious. So when you infect a mouse with the S strain, it dies. The R strain is not infectious. Okay, so when you infect the mouse with the R strain, it's not gonna die. Okay. So what we actually at this point we already know that when a virus, let me not make any assumptions. Let's just get straight into this. So the first step is we infect a mouse with S strain, it dies. We infect with R strain, it lives. Okay. Then he went on to do another test. He killed the S strains by hitting them, and after hitting them or after killing them, and infecting the mouse with the killed S strains, the mouse lived. The amazing thing is when he combined the killed S strains with the living R strains, the mouse died. So I think it raised a lot of questions, but I don't know if he didn't pursue this further. But another man did, or different men did. Avery, MacLeod, and McCarthy took it a step further. Okay, so what they did was they picked the killed S strain combined. So they are going to do a series of experiments over here. Okay, so in the first step they took the killed strains with no component of the killed strains destroyed, combined with the R strain, the mouse died. Okay, so now they are going to deduce whether carbohydrate or protein or lipid or RNA or which of the biomolecules present in the S strains are responsible for transferring genetic material from the virus to the mouse. Because we know when a virus infects its host organism, it's going to transfer its genetic material into the host. Okay, and start replication, and that's why the mouse dies. Okay, so what we are going to do is we want to know what the virus transfers to the mouse that makes the mouse die. All right. So the second one was he took the killed S strains, destroyed the polysaccharide or the carbohydrate components. The mouse died. Okay. He actually mixed them with the R strains as well. He did same for lipids. Mouse died. RNA. Mouse died. Proteins. Mouse died. But when he did same for DNA, when he destroyed the RNA component, the mouse lived. Okay. So this actually goes on to tell us that DNA is the genetic material, or the virus is transferring DNA to the mouse, and that's what is called causing the mouse to die. So it is DNA that's the genetic material, and not protein, nor RNA, nor lipids, nor carbohydrates. Okay. So Hershey and Chase also did another one. Okay, and this a bit interesting. Okay, the experiment used radioactive labeling to track what part of a virus enters the bacteria. Okay, so they wanted to know if it was the protein part of the virus that entered the host organism, or the DNA part. Okay, so they actually worked with bacteriophages. So if you hear a phage is a virus that can infect a bacteria. Okay, so they worked with a phage called a T2 phage, and the host organism was E. coli. Okay, so why did they use radioactive labels? Okay, so we know that proteins contain sulfur and DNA does not. Okay, and also DNA contains phosphorus, and most proteins or all proteins have very little phosphorus in them. So if you are going to use like two labels like sulfur 35 and phosphorus 32, we know that if we do that, sulfur 35 is going to label the proteins, and because DNA does not have sulfur, okay, and phosphorus 32 is going to label DNA because most proteins have little phosphorus, okay. So in the first part of the experiment, which is this side, the T2 phages were grown in a medium containing the radioactive sulfur, okay, and as established earlier, this is going to label the protein coat of the phage, okay. So these labeled phages infect when we infect the phages when we infect the E. coli with the phages, the mixture was put into a blender, blended and blah 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 blah. So after the mixture was put into a blender, the blending or centrifugation thing knocks off the phage protein coats, okay, from the bacterial surface, or you can see the phages infected the E. coli. Okay, so what happened is, if the E. coli, the phage actually infect the E. coli, is going to insert some part of it into the E. coli and some part will fall off. So we are just doing this to determine which part of the virus goes in and which one stays outside. Okay, so after the blending part, we expect that particular reaction or that particular process to have gone to completion. Okay, the part where the phage inserts some part of it into the E. coli and some part of, okay. So what they did was, most of the radioactivity was found in the phage ghost, that's like outside the E. coli cell, okay, and not in the E. coli cell. So the conclusion from this particular one is like the proteins, which were the labeled ones, those here, okay, does not enter the bacterial cell. So when a virus infects a host organism, the proteins does not enter the host cell, okay. Hence, protein is not the genetic material, okay. So they did the second part, and that's where they actually repeated the same thing but with a different radioactive label, which was phosphorus 32, okay. They did the same thing, infected E. coli, blah blah blah, after blending and centrifuging, and after observation, they realized most of the radioactivity was found inside the bacteria, okay, and this time around not outside. That means, and we know phosphorus label DNA, and if you are finding the phosphorus label inside, that means DNA actually enters the bacteria, the E. coli, okay. Hence the DNA carries the genetic information needed to produce new phages, because we know when a virus infects a host organism, is going to need the host to reproduce, okay. So hence it is the DNA that carries the genetic information needed to produce new or to replicate, okay. So this is just a summary of the experiment Hershey and Chase did or conducted. Okay, let's look at the central dogma of molecular genetics as well. This is not really a big deal. Central dogma is just three should I say processes, okay, that okay, from DNA replication to protein synthesis, okay. So DNA replication, transcription and translation, these three come together to call what we call the central dogma of molecular genetics, okay. DNA replication is simply the DNA unwinding into two strands, or you can call it unwinding into two daughter strands, okay. And transcription is when a DNA unwinds, we can use one of the strands as a template to transcribe an mRNA, okay, and that mRNA will go on to be translated into a protein, okay. So from the DNA unwinding, being transcribed into mRNA, and the mRNA being translated into protein, is what we call the central dogma of molecular genetics, right? So let's look at this particular slide as well, okay. And this particular page and the next, okay, let's look at this one first. This is just talking about the differences in DNA regulatory sequence, okay, not the protein itself, but the differences in DNA regulatory sequences can explain phenotypic differences like whether adults can digest milk or not. Okay, so if you look at this diagram here, what the diagram shows is a lactase gene. So this whole thing you see here is a lactase gene. Okay, it's actually labeled here. So a lactase gene, we know a lactase gene codes for enzyme lactase, and the lactase is responsible for digesting lactose, which is a milk sugar. Okay, so in humans, or in most humans, we know lactase is expressed in infants because basically infants take milk, right. So when we are growing, these lactase genes are actually turned off. Okay, but in some populations the lactase remain active. Okay, so in humans with these genes turned off, when they grow and they take milk, they tend to be lactase or lactose intolerant, right? Because they don't have the genes producing the necessary enzymes to digest or break down this lactase in the lactose in the milk, right? But unfortunately, fortunately, most of us now have our lactase gene still turned on. Okay, that's not normal, but I mean, if most of the populations are abnormal, it will be the new normal. So people with lactose intolerance are now seen as sickless or something. Okay, so let's look at the regulatory element. Okay, and not the coding region. The protein coding sequence of the lactase gene is largely the same in everyone. Okay, but the difference in those who are lactase persistent and those who are lactase intolerant are the is in the regulatory DNA element upstream of the gene. Okay, so if you look at this blue regulatory element, you realize in different populations they have different sequences. It will be like an SNP, like a single nucleotide, just a single nucleotide polymorphism. Okay, so and in Arabia, for example, where there is supposed to be a T, they have a J. In Europe, there's supposed to be a C, they have a T. Ethiopia, blah blah. Okay, so actually the gene in all humans are the same, but the regulatory sequence in different populations are different, and this determines whether the gene will be turned on when you grow or turned off when you grow, right? And the regulatory sequence actually is bound by a protein or regulatory protein called the OCT1. Okay, so this OCT1 helps recruit the RNA polymerase complex and controls whether transcription happens in adults or not. Okay, so we can talk about the SNPs. As I said, Arabia have different nucleotides, single nucleotides like changes. Okay, in different populations. So these SNPs, which are the variations in the regulatory sequence, improve the OCT1 binding and the regulatory elements. Okay, and they keep the lactase genes turned on in adults. Please, sorry for the background noise. I do not stay alone. This hostel is a bit noisy. So let's look at the next one. So so I think it looks like kind of messy, right? But it's not really that of a big deal. So what kind of graph is this? It's just like a Manhattan plot or something. Okay, but what we know is it's just using genome-wide association studies. Okay, so this particular diagram is just some researchers trying to compare Tibetans and Han Chinese. Okay, so from this diagram you can see they tested millions of SNPs across 22 human autosomes. So 1 2 3 blah blah to 22. Okay, so you can ask which SNPs differ significantly between the two populations, but we are not really going to focus on this. Okay, this is just to show the difference between like some populations. Okay, I'm not going to waste time on this. And as I said, please finish your term paper on time. Okay, I'll see you in the next one.